Good morning, students. Today we will be discussing about immunology, the immune system, the basics of infection, how the complement systems will be activated, and complement cassette system, all those things we'll be discussing in the first unit. So your first unit deals with infection and complement system. So the basic term immunity, which means the defense system that has evolved to protect animals from invading both pathogen on pathogenic microorganism and also the cancer cells. So together we will be calling it as immunity, and the study of the immune system, you call it as immunology. So most of the times the invaded pathogen, not only the invaded pathogen, but also any of the cancer cells which are occupied or which are invading our biological system, automatically that will be cleared off. So the most important function of the immune system, which will be two, one is recognition and the response. So recognition of the self cell will be avoided, and the recognition of non-self cell will be considered as the immune response. The immune response will be initiated only for the non-self cells. So the first thing is your immune system will recognize whether those cells are self cells or non-self cell. If it is a pathogenic particle or any viral particle which enters, the first thing what your immune system will do is it will first check whether it is your own cell or it is invaded pathogen. In the same manner, the cancer cell also will be recognized whether it is a normal cell or it is so different from your normal cell or your cancer cell. So the response will be of two types or two ways the immune system will respond. One is through humoral immunity. The other one is through cell-mediated immunity. So through humoral immunity, immunoglobulins, which we call it as antibodies will be produced, and those antibodies will go and neutralize the antigens. Whereas if it is cell-mediated immune system, all the phagocytic cells, which includes blood monocytes, macrophages, which will be in tissues, and neutrophils, all these cells will go and perform phagocytosis process. Thereby, it will clear off the invaded pathogen. So there are two types of lymphocytes which will carry both humoral as well as cell-mediated immune response, out of which is T lymphocytes. It is derived from thymus gland, which mediates the cellular immunity. Whereas the B lymphocytes, which will be derived from Barossa fabricus, which will mediate the humoral immunity. So the B cells only will be producing the plasma cell, and the plasma cell will be producing antibodies. So according to the clonal selection theory, an individual lymphocytes which expresses a membrane receptors that are specific for a distinct antigen. This unique receptor specificity is determined before the lymphocyte is exposed to the specific antigen. Binding of antigen to its specific receptor activates the cell, causing it to proliferate into a clones of cells that have the same immunological specificity as the parent cell, so that the clonal expansion will be happening, so that if more of the same kind of pathogen which is encountered, automatically the immune system will be capable of clearing all those pathogens. Innate immunity, which provides the first line of defense against infection. Adaptive immunity responds to the challenge with a high degree of specificity as well as the remarkable property of memory. So only in the adaptive immune response, once the infection happens, the same kind of infection, if it reoccurs, you will not get the symptom. Instead, the pathogen will be cleared off by means of the memory cells. So first we'll start with the non-specific host defense mechanism, which includes our anatomic barrier. The first line of defense will be the skin's mechanical barrier, which retards the entry of microorganism, which has acidic environment of pH 3 to 5, which retards the growth of microorganism. Then we have mucous membrane. The normal flora competes with the microbes for the attachment sites and nutrients. The mucus entraps foreign microorganism, and the cilia propels microorganism out of the body. Then we have physiological barriers, which includes first temperature, the normal body temperature, which will be inhibiting the growth of some pathogens. In case if we have fever, which is a symptom of a particular infection, automatically this fever will raise the body temperature by 1 or 2 degrees, which makes the particular microorganism, invaded microorganism, to be destroyed. So for temperature is the first defense mechanism. Then low pH, acidic pH of stomach content kill most of the ingested microorganism. So the saliva itself will clear off maximum microorganism which we intake through food or while speaking, whatever the pathogen which enters. And in case if it crosses the salivary secretion, automatically if the pathogen enters into the stomach's acidic pH will automatically kill the ingested microorganism. So apart from that, we also have chemical mediators, which includes lysozyme, which cleaves the bacterial cell, thereby it just clears off the bacterial infection, interferons, which includes which induces antiviral state in the uninfected cells, so that the uninfected cells will be protected from the viral infection to complement lysis microorganism or facilitate phagocytosis mechanism. Then toll-like receptors, which recognize the microbial macromolecules, which signals the cell to secrete immunostimulatory cytokines. One example is collectins, which disrupt the cell wall of pathogen, invaded pathogen, automatically thereby it destroys the pathogen. Then we have phagocytic and endocytic barriers. Various cells internalize and break down foreign macromolecules. Specialized cells, which includes blood monocytes, neutrophils, tissue macrophages, internalizes and kills and digests the whole microorganism through phagocytosis mechanism. We also have inflammatory barriers. Tissue damage, infection induces leakage of vascular fluids, which contains serum proteins with antibacterial activity and influx of pathogenic cells into the affected area. So pathogenic cells, which will be invading and it will be performing phagocytosis mechanism and it will clear off the pathogen. So we have different types of infections, which includes viral infection, which is caused by the virus, either DNA-containing virus or RNA-containing virus. Then we have bacterial infection, which will be by means of different varieties of bacteria. Then we have fungal infection by means of different fungi, parasitic infection by means of different parasites. Then we have transmissible spongiform encephalopathies, which includes our prion disease. So these are the different types of invading pathogens which cause infections and allergic responses. So immunogenicity is the ability to induce a humoral or cell-mediated immune response. So if B cell is recognizing any antigen, the invaded antigen, automatically the B cell will produce effector cells. The effector cells, along with that, we will also be able to produce B cells. So the effector or the activated B cells will produce plasma cells, and the plasma cells in turn will produce memory cells. The plasma cells will be producing antibodies, and the antibodies will be going and destroying or neutralizing the antigen which is recognized by the B cell. So whatever the B cell which has recognized the particular antigen, the plasma cell which produces the antibody, that antibody will go and neutralize the recognized antigen. And the same way, the T cells, if it is recognizing the antigen, effector T cells will be produced along with the memory cells. And the effector T cells also will be producing the cytotoxic T lymphocytes, then T helper cells and all. And these cells will go and neutralize the antigen. So antigenicity is the ability to combine specifically with the final products of the above responses. All molecules that have the property of immunogenicity also have the property of antigenicity. Some small molecules called haptens are antigenic but incapable by themselves of inducing a specific immune response. They lack immunogenicity. Immunogenicity is not an intrinsic property of an antigen, but rather depends on number of properties of a particular biological system that antigen encounters. So let us see now the nature of infective agents, how and what are the ways the infective agents will be very effective in causing the infection. The nature of infective agent depends on whether it is foreign, so foreignness based on the molecular size, chemical composition, the complexity, and the ability to be processed and presented with the help of MHC molecules on the surface of antigen-presenting cells or altered cell cells, which includes the cancer cells. So foreignness, if the particular cells are very foreign, automatically the immune system will react. First, it will recognize and then automatically it will respond to those foreign antigens or foreign particles or the altered cells and all. So the self component like corneal tissue and sperm, these two are sequestered from the immune system. So automatically these two, if you inject, automatically these two will react as immunogens. Otherwise, all the biological cells, the normal cells will be considered as self cells. So only the immune response will recognize the non-self cells or non-self particles. So non-self particles are very effective infective agent. The molecular size, most of the active immunogens tend to have a molecular mass of 1 lakh Dalton. If it is less than 5,000 to 10,000 Dalton, they are very pure immunogens. Also, there are few substances which has molecular weight less than 1,000 Dalton have proven to be immunogenic in nature. It is based on the chemical composition and also the complexity of the particular component or the particle. So the chemical composition and the complexity also will determine the nature of the infective agent, which includes the size and foreignness. Not only that, the complexity also. So the polymers, which are homopolymer in nature, which is made up of same amino acid, same sugar, those kind of polymers will lack immunogenicity regardless of their size. If the size is also too big, as we discussed, if the molecular size is larger, automatically it will be immunogenic in nature. But though they are larger in molecular weight, homopolymers will lack immunogenicity. Whereas the copolymers, which is composed of different amino acid, different sugars, all these are immunogenic in nature when compared to homopolymers. So the study shows the chemical complexity will contribute for the immunogenicity. And we can use lipids as antigen too. For the stimulation of B cell responses, the lipids are used as haptens and attached to the suitable carrier. Molecules such as protein, keyhole, implet, hemocyanin, or bovine serum albumin are used. By immunizing with these lipid protein conjugates, it is possible to obtain antibodies that are specific for the target lipids. By using this approach, antibodies have been raised against a wide variety of lipid molecules, including steroids, complex fatty acid derivatives, and fat-soluble vitamins, such as vitamin E. Susceptibility to antigen processing and presentation. So large insoluble macromolecules generally are more immunogenic than small soluble ones because the larger molecules are more readily phagocytosed and processed. Macromolecules that cannot be degraded and presented with MHC molecules are poor immunogens. So overall, whatever the property we have discussed, all those properties will induce the immune response and certain properties will stop the